Hello, Assalamualaikum warahmatullahi wabarakatuh. Welcome back to Food Science Study Program, Department of Food Science and Technology, Faculty of Agricultural Engineering and Technology, IBB University. My name is Rati Dewanti Haryadi. I am a professor in food microbiology at IPB University. We are going to meet today to discuss our course ITP 322 Food Safety and Sanitation. We have come to our fifth lectures, and the topics for today's discussion is soil and biofilms. The topic will cover soils, type of soils in food industry, understanding soils, soils classification, and then we are going to also discuss biofilm, the definition, the advantage and benefit of biofilm in nature, biofilm formation on solid surfaces, and biofilms in food industry. When you completed this lecture, you are expected to be able to describe various soils in food industries, describe characteristics of soils, differentiate visible and invisible soils, describe steps in the biofilm development, and describes the advantages of biofilm formed in food industry. Let's start our discussion with soils. Soils in food industry is generally defined as unwanted matters on food contact surfaces. They can be visible, you can see it with your eyes, or invisible, you can't see it with your eyes. The primary sources of soil can come from the food product itself. Food products generally comprises of carbohydrate, fats, protein, and minerals, from water residues and cleaning materials residues, as well as biofilms formed by microorganisms. Soil's composition vary widely, thus no one detergent is capable of removing all types of the soils. Many complex soils contain combination of food components, cleaner components, as well as hard water salts. The film vary in their solubility, and it all depends on whether it has been exposed to the heat, dryness, as well as age. If you are a person responsible for developing a sanitation program in a food industry, you must have a good understanding of the nature of the soil. Improper use of detergent can set soils, making them more difficult to remove. For example, if you use acid cleaners for example, protein soils, you may end up making the protein soil denature and making them more difficult to be cleaned. Many films and biofilms require more sophisticated cleaners, usually by adding oxidizing agent. And as a rule of thumb, usually we say that acid cleaners dissolve alkaline soils, while alkaline cleaners dissolve acid soils. So, soils in food industry usually is classified based on its solubility. Visible soils can be classified as soluble in water. Those include sugars, some starches, and most salts. Soluble in acid, limestone, and most mineral deposits. Soluble in alkaline, such as protein and fat emulsions. Some of the soils, complex one, may be soluble in water, alkali, or acid. The physical condition of the soil deposit also affect its solubility. Freshly precipitated soils in a cool or cold solution is usually more easily dissolved than old, dried, or baked-on deposits or a complex film. Let's start with carbohydrate-based soils. Simple sugars are generally easily soluble in warm water and quite easily removed. Starch residues are also easily removed with mild detergent, but when starches associated with protein or fats, they will require a highly alkaline detergent. Fat-based soils is present in an emulsion and can generally be rinsed away with hot water, but more difficult fat and oil residues can be removed with alkaline detergent with a good emulsifying or saponifying ingredients. Protein-based soils are the most difficult soil to remove in food industry. They range from simple proteins, which is easy to remove, to more complex protein, which is very difficult to remove. Protein that is heat denatured can be extremely difficult to remove. Generally, a highly alkaline detergent with baptizing or dissolving properties is required to remove protein soils. In addition, wetting agents can be used to increase the wettability and suspendability of all proteins. Protein films, in general, require alkaline cleaners with hypochlorite and wetting agents. Mineral salt-based soils can be either easy to remove or troublesome deposits or films. Calcium and magnesium involved in some of the most difficult mineral films. Under heating and alkaline pH, this mineral can combine with bicarbonates to form highly insoluble complexes. This is also true for deposits containing iron or manganese, and salt films can cause corrosion of some surfaces, and difficult salt films require acid cleaners, especially organic acids. Sequestering agents such as phosphate or chelating agents are often used in detergent for salt film removal. This table shows you a variety of soils that commonly encountered in food industry, their solubility, easiness to remove, and the changes in terms of easiness to remove when they experience heating. Now, what about invisible soils? Invisible soils in food industry are resulted from microscopic organisms or microorganisms. They consist mainly of bacterial spores and to a lesser extent may contain also vegetative bacteria, fungal spores, or yeast, and they inhabit unclean sites, cracks, and pits in the surfaces of equipment, walls, or ceilings. These invisible soils are subject for environmental monitoring. Microorganisms then can attach to solid surfaces and form biofilms. What is biofilms? Biofilms are a collection of microorganisms, mainly bacteria, that grow together in a matrix of polymers called extracellular polymeric substances produced or secreted by themselves on solid surfaces. This EPS forms on surfaces and builds up over time, allowing spoilage and or pathogen to flourish and lead to contamination. When it comes to biofilm, we should know the terms that are commonly used. Sessile cells, those are referring to cells that are attaching to surfaces, while the cells that are growing or living in a liquid phase is called planktonic cells. These are the graph of biofilms. On the left, you can see Bacillus cereus biofilm on a stainless steel surfaces. And on the right, you can see E. coli 015787 biofilms on the similar surface. In nature, biofilm actually could benefit for human being. Naturally formed biofilm in the sediment in the bottom of a body water, such as river or ponds, is known to reduce pollutant in water. They can be used in a trickling filter system for wastewater treatment. They also commonly used to extract minerals and metal in mining industries, and they are simulated in fermentation technique to form immobilized bacteria or immobilized microorganisms to be used in fermentation. However, there are also disadvantages caused by biofilm. They have been reported to cause infection due to implanted medical devices. They cause falling in ship hulls, in equipment of industry, clogging in a wastewater system, and a potential source of contamination in drinking water distribution system, such as pipes, as well as a potential source of contamination in food industry. So the question is, why do bacteria form biofilm? There are several theories. One, bacteria growing in a low nutrition undergo changes in their cell membrane structure, thus becoming more hydrophobic. When these hydrophobic cells attach or stay close or in close proximity to hydrophobic surfaces, they may be interaction and they attach. Secondly, low nutrition also causes bacteria to experience size reduction and also becoming more hydrophobic. You can see this phenomenon in dwarfing in Vibrio and rounding in E. coli 015787. When the cells become hydrophobic, they also bind to each other or attach to each other, forming aggregates with a higher mass. Thus, gravity can play a bigger role, causing them to attach to surfaces. The slide shows you steps in biofilm formation on solid surfaces. Adsorption of organic matters by solid surfaces may affect bacterial attachment to solid surfaces. Transport of bacteria from liquid phase to surfaces may follow, and when the adsorption occur, it may end up with reversible initial attachment. The reversible initial attachment can proceed to irreversible interaction of bacteria and the surface when there is a stronger chemical reaction such as hydrophobic interaction or ionic interaction. The irreversible interaction can proceed to the growth of bacteria on the surface, followed by production of extracellular porous saccharides, and it may also result in some detachment of bacteria from the surfaces, but the net product is bacterial accumulation to yield biofilm. This graph shows you the reversible attachment that occur in the beginning of a biofilm formation, followed by irreversible attachment, followed by growth and multiplication, extracellular polysaccharides formation, some level of detachment, and at the end you will have a accumulation of bacteria on the surfaces. Biofilms pose some problems because bacterial biofilms or sessile cells are more resistant to adverse conditions such as heat, dryness, antimicrobial agents such as antibiotics, disinfectant, or sanitizing agents. Therefore, it is reported that antibiotic treatment in patients with implanted devices, as well as cleaning and sanitizing in hospital or food industry, may need modification or adjustment. So why are biofilm bacteria more resistant to biocides or antimicrobial agent? First, because they are indeed protected by the polysaccharides that they produce themselves. Secondly, because they are protected, only smaller areas are exposed to the antimicrobials that we apply. In addition, lower growth rate of biofilm bacteria is similar to stationary phase planktonic cells, which is generally more antibiotic or antimicrobial resistant. Now, in food industry, the possibility of biofilm formation are significant because they have all the factors needed for biofilm formation. For example, liquid phase, solid surfaces, nutrients, and microorganisms. Biofilms has been reported in the floor, walls, pipes, and other surfaces such as table, gasket, conveyor belt, slicer, etc. in food industry. How do biofilm bacteria contaminate products? One is because of the residue is not thoroughly clean, thus they become the source of contamination. Secondly, because of the detachment of some biofilm fragments that are transported to other part of the facility. The following slide shows some experiments on biofilm. The slide shows biofilm formation by Salmonella and Chronobacter sakazakii on 3 different surfaces. In general, you can see that at 24 incubation, biofilm has been formed in all surfaces with a density between 10 to the 4th to 10 to the 5th. Out of the three surfaces, density of biofilm are usually found more in plastic surfaces, followed by rubber, and then stainless steel. The slides show you the effect of several sanitizing agents to planktonic and biofilm. As you can see here, we started with the biofilm or planktonic cells with the initial count of 10 to the 6 or a million per mil or per centimeter square that can be inactivated fully by chlorine, cause 4 log reduction by quartz, but iodophore can kill 5 log of the planktonic cells but none of the biofilm, suggesting that biofilm bacteria are more resistant to iodophore. With the older planktonic and biofilm, you can see some changes, still the same thing with chlorine, but you can see the difference in the effect by quartz as well as by iodophore. In these experiments, we would like to know whether a surface with biofilm that has been inactivated using chemicals or cleaners can promote a reformation of biofilm. So when previous biofilm was inactivated with sodium bicarbonate or by chlorine, you can see biofilm formation is well promoted. But when you add some brushing to the first biofilm, then the next biofilm formation are less. These are several approaches proposed for biofilm mitigation. So ideally, preventing biofilm is the best, a more logical option than treating after biofilm is formed. When the biofilm is formed, then you have an option of cleaning and sanitizing using a reformulated chemicals. Or we can also do some surface modification using surface materials that do not promote attachment or by supplementing it with some nutrients that will deter bacterial attachment. In addition to reformulation of cleaners and also sanitizing agents, cleaning and sanitizing of biofilm can be improved by increase of exposure time, increased temperature, and the use of mechanical activity such as brushing. Here are several sanitizing agents that are produced commercially that has been proven to be able to inactivate biofilm. And my last slide showed you several combined disinfectants that are recommended to be used to eliminate a certain form of biofilms. Thanks so much for participating in this class. We are looking forward for the next material. Don't forget to look at the materials available in the LMS. And Assalamualaikum warahmatullahi wabarakatuh.